This model is to show cellular respiration, the most efficient and prevalent catabolic reaction. This black line represents the cell membrane, a phospholipid bilayer. Inside the cell membrane would normally be many organelles, but here, drawn not to scale, is one mitochondria with its two phospholipid bilayer membranes, the outer one in green, the inner one in purple. These many folds increase the surface area and are called the cristae. This area inside is called the mitochondrial matrix, and the pink area in between the two membranes is referred to as the inner membrane space. Cellular respiration is a combination of 3 steps, beginning with glycolysis, the Krebs cycle, and ending with oxidative phosphorylation. Glycolysis begins when a molecule of glucose moves into the cell. Through a 10-step process, that molecule of glucose is broken down into 2 molecules of pyruvate. In doing so, 4 molecules of ATP are released. However, 2 ATP molecules must be invested into the early stages of glycolysis, leaving us with a net gain of 2 ATP. 2 water molecules are also released, as well as 2 molecules of NAD+ are involved. These act as electron acceptors and are reduced when they accept the electrons. 1 molecule of NAD+ accepts 2 electrons and becomes 1 molecule of NADH plus a hydrogen ion. At this point, glycolysis is complete and the 2 molecules of pyruvate move on. Only a quarter of the energy of glucose has been harvested at this point, with the net gain of 2 ATP and the 2 molecules of NAD+ being reduced. Pyruvate is now ready to move into the mitochondria and does so via a transport protein and active transport. As pyruvate moves in, another molecule of NAD+ accepts 2 electrons and is reduced to NADH. A carbon dioxide is given off, and what is left of pyruvate combines with a coenzyme A to form the molecule known as acetyl-CoA. Since glycolysis, 1 molecule of glucose yields 2 pyruvates. This happens twice. Another molecule of pyruvate moves in, another NAD+ is reduced to NADH, another molecule of carbon dioxide is given off, and another acetyl-CoA molecule is formed. These acetyl-CoA molecules are what move on to the next step, known as the Krebs cycle. As they move through the Krebs cycle, the following things occur. A molecule of water must be spent, a molecule of carbon dioxide is given off, 3 molecules of NAD+ are reduced to NADH, and FAD, another electron acceptor, accepts 2 electrons and is therefore reduced to FADH2. Again, since glucose yields 2 pyruvates and therefore 2 acetyl-CoAs, the Krebs cycle spins through again with the second molecule of ATP, and we get double these products. Additionally, there is a net gain of 2 ATP from the Krebs cycle, 1 from each rotation. At this point, most of the energy from glucose is tied up in these electron carriers of NADH. These are the molecules that are going to move on to the final stage known as oxidative phosphorylation. Oxidative phosphorylation begins with the electron transport chain, a combination of proteins and prosthetic groups located in the inner membrane of the mitochondria. NADH, or FADH2, brings the electrons to the electron transport chain. They drop the electrons off and are oxidized back to NAD+, as well as a hydrogen ion. These electrons that are dropped off in the chain are pulled downhill by increasingly electronegative ions until they finally reach a molecule of oxygen. This happens over and over again with all the electrons being carried by NADH. FADH2 can also drop off its electrons but drops them off at complex 2 instead of complex 1. The implications of this are, as NADH drops off its electrons, hydrogen ions are pumped into the intermembrane space. Because FADH2 enters at complex 2, fewer hydrogen ions are pumped into the intermembrane space than if it entered at complex 1. The final electron acceptor, like we said, is a molecule of oxygen. As it accepts these electrons, combining with hydrogen ions in the mitochondrial matrix, it is converted into a molecule of water. This buildup of hydrogen ions in the intermembrane space creates a hydrogen ion gradient. The inner mitochondrial membrane acts like a dam, holding back all this potential energy of a high concentration of hydrogen ions. The tendency here would be passive transport, for hydrogen ions to flow from an area of high concentration in the inner membrane space to an area of low concentration in the mitochondrial matrix. However, the only point in which they can passively flow back through the inner mitochondrial membrane is via a molecule called ATP synthase. Over time, molecules of spent ATP return to the mitochondria as ADP molecules. For every 3 hydrogen ions that flow back through ATP synthase, 1 molecule of ADP is converted to a molecule of ATP. Another 3 molecules come through, another molecule of ADP converted to ATP. Throughout cellular respiration, we received 2 ATP molecules from glycolysis, 2 ATP molecules from the Krebs cycle, and 32 to 34 molecules of ATP via the electron transport chain and oxidative phosphorylation. This gives us a total of 36 to 38 ATP molecules for every molecule of glucose that enters the cell. The end.